Hello, ladies and gentlemen. Mr. Hescox here to finish out the last part of Chapter 5, Section 3. Uh, we've been talking about selective breeding. One type of selective breeding is a hybrid. A hybrid is when you cross two different species that are similar, such as a cow and a buffalo, a lion and a tiger, you get a liger, cow and buffalo, you get a beefalo. If you do a donkey and a horse, you get a mule, or if in some cases you get a zonkey if you do a zebra and a donkey. So we're going to talk about humans manipulating DNA that we call genetic engineering today. Your homework is 5.3. Within the last 30 years or so, humans have began manipulating DNA to do exactly what we want. It's called genetic engineering. Basically, you either isolate a piece of gene from its in one organism, and you either insert it or cut it or put it back into another organism. So I've had a chance to do this in college. We took a gene from a jellyfish and made bacteria glow in the dark. In fact, the professor that made it called it Mr. Green Genes. Genetic engineering has been used a lot with our foods. It's been made all of our food more and more efficient. So we use less pesticides, less fertilizers because we've genetically engineered our crops to need less fertilizer, to need less pesticides. One such way, they made Roundup resistant corn. Well, Roundup is a weed killer. They've genetically engineered corn so that when the corn starts growing, they spray the top of it now with Roundup. All of the weeds die and the corn gets all of the nutrients so it doesn't have to compete with the other things. We've genetically engineered tomatoes and cabbage, different types of plants to grow in saltier or less, more specific soils. The big benefits of genetic engineering: more food, disease-resistant plants, and then the future of genetic engineering is still in medicine. And some of the medicines we don't even know yet, but with fixing you right now, we have growth hormones and insulin that diabetics need to stay alive. Those would not be possible without genetic engineering. Some of the risks to genetic engineering: some people are worried that the possible pesticides that we've engineered in might be damaging to humans. And there's always the hope that some people might develop a bacteria that we can't kill with antibiotics, and then it would be like the super bacteria. DNA and genetic engineering is very, very important to the police in crimes because your DNA is very specific to you. Unless you have an identical twin, you have regions of DNA that are very exact. And if your DNA or your blood splatter is on the wall, it's a pretty good idea that you were there. And many, many people have been freed from jail because DNA evidence says, no, that's not the right DNA. But in some cases, you might sit in jail for a while because they didn't have the technology to test the DNA like they do today. You can have your DNA read today. It's about $100. Ancestry.com has one, 23andMe, and there's another one about chromosomes that will read your DNA and tell you each gene that's in there. They'll tell you your background as far as ethnicities. They can even tell you diseases that might be in there. The largest genetic project ever created was the Human Genome Project. It was finished in 2002. They basically mapped or read every A's and T and T and G, A, T, C, and G in the human genome, all 6 billion. And this was a major accomplishment and has led to all of the things that we can do with genetics today. One of the first clones ever, as far as a mammal goes, is Dolly the sheep. Here she is. She did not live that long, like 6 or 7 years at the most. But cloning is very common in different types of bacteria. They've cloned cats. One of the weird things they've cloned in cats was they cloned a calico cat. And this calico cat, when they cloned it, was actually gray and white. The calico trait did not show up again in the clone. I've seen monkeys have been cloned, but currently today it is illegal to clone humans. Now, it is important to clone things like bacteria because that's where we get the insulin that our diabetics need. Now, the simplest type of genetic engineering is called recombinant DNA. Basically, bacteria have very simple DNA. They don't have a nucleus. So they have these little pieces of DNA that are circle, and we can actually use enzymes to cut that circle, insert our new gene in, and then you just reinsert these plasmids back in, and it looks something like this. So there's the gene for, this is may say insulin. You take this plasmid ring, which is just a circular piece of DNA, and you cut these sticky ends into it. And then you use a type of enzyme that glues it back together called DNA ligase. We insert it back into the bacteria and let the bacteria live. And the bacteria won't know that this DNA is not supposed to be there, and they read it and they make insulin. We capture and purify the insulin and give it to diabetics like my sister or like many of the other kids who have been to school, and it keeps them alive. Some of the major products, insulin, growth hormone, different types of vaccines are genetically engineered. Interferons are actually specific proteins that are to block viruses. Um, and then in agriculture, we use for like frost resistant, disease resistant plants, bacteria resistant, insect resistant, all of them either allowing people to live longer or have more food, which sometimes can cause people to live longer too. Ladies and gentlemen, that takes us to the end of our chapter. Sooner or later, we will be having a test and a quiz. I wish you all well. Make sure you get your homework done, and I'll see you next time. I'm Mr. Hescox, signing out.